In this video, we're going to talk about tRNA and its function in the process of translation. First, let us travel back in time to before the discovery of tRNA. We know that the central dogma described the flow of genetic information from DNA to functional products such as proteins. First, the genetic information from DNA is copied into a new single-stranded molecule called messenger RNA in a process called transcription. Next, the genetic code in mRNA is decoded to produce a sequence of amino acids in a polypeptide chain in a process called translation. Before the discovery of tRNA, Francis Crick had hypothesized that there must have been an intermediate molecule that functioned to help with the conversion of the genetic code from mRNA to a sequence of amino acids. He called this mystery molecule the adapter molecule, and we will soon see why such a name was appropriate by looking at the function of tRNA. When you look at the structure of tRNA, you will see that there are two major areas, a trinucleotide area called the anticodon and an amino acid attaching site. Amino acids are the basic building blocks of proteins. A tRNA molecule can have many different anticodons, and each anticodon will determine the specific amino acid that is attached to the tRNA by following the genetic code. During translation, the first tRNA molecule binds to the mRNA at the complementary base pair attaching site with the help of a ribosome that consists of a large and small subunit that come together around the mRNA to help drive polypeptide synthesis. Next, a second tRNA molecule carrying another amino acid attaches to the mRNA at the complementary base pair attaching site, and the two amino acids link together to form a peptide bond. The first tRNA molecule detaches from the mRNA, leaving behind its amino acid, and the mRNA then shifts through the ribosome. This allows the next tRNA to attach to the mRNA and bind its amino acid to the adjacent amino acid, forming the polypeptide chain. This process will continue by adding amino acids to the polypeptide chain until it reaches a stop codon, at which point the desired product, which is usually a protein, is formed. After describing the function of tRNA, it is easy to see why it is referred to as the adapter molecule. Since you can't connect an amino acid to an RNA template directly, you need to have a tRNA acting as an intermediate. As an analogy, say you have a camera of photos you wish to transfer to your computer. The photos in the camera are stored in an SD card, which is not immediately compatible with your laptop. Instead, you need the help of a USB adapter to exchange the information from your SD card to your computer. In this case, the information stored on your SD card is like an mRNA sequence, and the USB adapter is like the tRNA that allows you to transfer that information to your computer. But what if there was a problem with the tRNA that caused it not to follow the genetic code? For example, what if proline attached to a tRNA molecule with a nucleotide sequence that was supposed to code for valine? Well, during polypeptide synthesis, anywhere there was supposed to be a valine will now be a proline. This could result in a non-functional protein. Depending on the importance of that protein, it can result in the death of the cell that contained the non-functional tRNA.